Okay, so let's get started. So our first speaker this morning is Dave Unwin. He's going to be talking about a reptilian mode of reproduction in pterosaurs, implications for pterosaur paleobiology.
Good morning. Thank you very much, Richard. So this is reproductive behaviour in pterosaurs, Mesozoic flying reptiles. This is how much we knew about reproductive behaviour in pterosaurs prior to 2004. Happily, since then, there have been a whole series of finds, these are a couple of them, which have provided us with some really important insights into the reproductive modes of pterosaurs. But we're dealing with pterosaurs, so inevitably there's been lots of controversy, there's been confusion, attempts to kind of depict what people believe to be going on often contain all sorts of errors and misconceptions, and much of what you see here is basically wrong, in particular this one, which if I have time, I'll come back to at the end. So the question might be, what do we really know about pterosaur reproductive biology? And the first step really is to sort of have a look very quickly at what the fossil evidence is for this particular behaviour. This includes Mrs. T, an example of Darwinopterus, which is a pigeon-sized pterosaur from the Jurassic of China. And the important thing here, this is a female associated with an egg, which is extremely useful in terms of understanding key aspects of reproductive biology. We also have two beautifully preserved embryos and eggs from the Yixian Formation of Liaoning, China. These are probably ornithocheirid pterosaurs. We have this absolutely extraordinary find published a couple of weeks ago in Science, which is an accumulation of more than 300 eggs, pterosaur eggs, amongst which there are some absolutely beautifully preserved examples, 3D examples, and around about 16 embryos. So again, this is telling and will continue to tell us lots of very interesting things about pterosaur reproduction. Finally, we also have a beautifully preserved embryo in remains of an egg of Pterodostro, the filter-feeding pterosaur from the Lower Cretaceous of Argentina. If we put together all the records we have of eggs and embryos, hatchlings and early juveniles shown in red here, calibrated against a phylogeny, we have a modest pterosaur fossil record of early growth stages, but one that's really, really useful in terms of understanding what's going on. And I've just shown on the left-hand side here various hatchling individuals and an egg there as well. So what I want to do in this very brief talk, as it turns out, is look at 5 aspects of pterosaur reproductive biology. Don't worry about reading all these because we're going to go through each one in turn. The context for this is phylogenetically is a group called Sauropsida, and Sauropsida basically is what you might call traditionally reptiles, which is this lot here, and birds. And the important thing here is that all these groups shown in bold have extant representatives where we have some understanding, sometimes not so great, of their reproductive modes, what they actually do. So these provide a really important context for understanding pterosaurs, but also they help us to understand what's going on there as well. So most sauropsids, you've just seen them, actually have paired ovaries. The one exception are birds, highly derived sauropsids which have a single ovary. Mrs. T seemed to suggest with her single egg that pterosaurs also may have only had a single ovary, which would have been an interesting example of convergence, except that when the counterslab was reported a couple of years back, we found not only the reverse of the original egg, but there's a second egg preserved in the body. I've had a look at it and I can completely confirm this. So paired ovaries in Darwinopterus seems extremely likely, and because reproductive systems generally are very conservative, I think it's highly likely that paired ovaries were probably the norm for pterosaurs. The relative size of the egg compared to the mother tells you something about how much parental investment there is in that particular group. Basal sauropsids tend to have relatively small eggs, relatively small investment. Derived sauropsids, i.e. birds, have relatively large eggs, quite a large investment. Mrs. T is very helpful here again because we have an example with eggs preserved with the adult. There are several ways of calculating the mass of Mrs. T, and it's a relatively straightforward exercise to calculate the original mass of the egg. You just use this expression here, and hey presto, it turns out 6 grams. So the context then is this. Here we see initial egg mass, so that's the egg when it was first laid, and here's the female mass, and this is for a large number of species of sauropsids. And you can see quite clearly birds have relatively large eggs, whereas reptiles, that group down here, have relatively small eggs. Where does Mrs. T fit? Well, she comes about there. Pterodostro comes in around about here. They've quite large ticks on here because our estimates of the mass of adults show quite a lot of variation. Hemipterus, that egg accumulation just been looking at, comes in somewhere about here, but I haven't finished sort of sorting that one out yet. But there are 2 main conclusions we can draw. First of all, pterosaurs seem to have relatively small eggs. And secondly, insofar as you can draw any conclusion from 3 points on a graph, yes, of course you can, Richard, the allometric slope seems to be more comparable to that of things like crocs and turtles than it does to birds. How significant is that? We will see in the future. How big was the clutch in pterosaurs? Well, this extraordinary accumulation from Hami in Xinjiang provides us some clues. It doesn't provide us with a concrete number because so far we don't actually have any nests, i.e. egg accumulations themselves, but it's very hard to imagine this accumulation forming from literally hundreds of nests in which there are just 1 or 2 eggs. I think this accumulation rather says nests have 5, 10, 15 eggs in there, maybe more. I'm confident at some point we will find nests in this deposit. The nature of the eggshell tells you very important things about the incubation environment and how that particular group incubates their eggs. Pterosaurs lay parchment-shelled eggs. We see that beautifully in the Xinjiang material. Here's that crumpled and folded up parchment-shelled egg of Hemipterus. It's identical to what you see in lizards and snakes when their parchment-shelled eggs dry out. There is a very thin, literally paper-thin, 60-micron thick calcareous layer that is found in Hemipterus. It's also present in Pterodostro. You see that how incredibly thin it is here in this break here. But that layer is entirely absent, for example, in the Yixianornithochirids and also in the Darwinopterus eggs as well. How do these eggs then compare with known eggshell types? Well, they're completely unlike the rigid shells that you find in birds or in crocodilians. For one thing, the shell's much, much thinner. They're a bit more similar to turtles, but here also the eggshell's quite well organised in terms of those crystalline units and is relatively thick compared to pterosaurs. Pterosaurs compare almost identical in many respects with the parchment shells of lepidosaurs, including these things like the Tuatara sphenodon, which also often have this very thin sort of calcitic layer over the top of the shell. What does this tell us about the nesting environment? It's very difficult to sit upon a parchment-shelled egg. It means almost certain that they've incubated either in vegetation or soil or sediment, quite possibly the sediment that you see that forms the deposit in Hami and Xinjiang. If that's correct, and it's very, very likely, for one reason being that parchment-shelled eggs dry out very, very easily, then that tells us that these eggs developed with their embryos at ambient temperatures, which means they probably developed quite slowly. Fifth aspect, if we look at sauropsids as a whole, they show a very wide range of behaviours and abilities when they hatch out. So on the one hand, we have hatchlings of lizards and snakes and crocs, which are highly precocial. Essentially, they're like small versions of the adults, except they can't reproduce, and they don't need any kind of parental care. On the other hand, if we go and look at some of the birds, particularly large birds, large seabirds, they require lots of parental care, and they're highly altricial. how do we figure out whether pterosaurs were precocial or not? In this case, we've chosen to focus on the flight ability, and I'll start by looking at ossification patterns. The embryos and hatchlings of pterosaurs are remarkably well ossified, far more so than the hatchlings of things like the starling, and much more like the hatchlings of extremely precocial birds like the button quail. And noticeably in pterosaurs, you already have this highly elongate forelimb is well formed, unlike that of birds. Wing membranes are present in lots of hatching individuals. Wing membranes preserved all over here in this specimen from the Yixian Formation. And if we look closely, it even has critical structures such as the actin of fibrilli, the organization, the fine detail of this is exactly as in adults. So they certainly have the equipment to be able to fly. When they were hatchlings, could they actually do it? It's a relatively straightforward task to produce reconstructions of the skeleton and the outline of the wing membranes, and to calculate very simple sort of aerodynamic features of these animals, exactly as we would do for adult pterosaurs. And in this case, what we find, if you plot wingspan here along the bottom against wing loading, this embryo, bear in mind it's an embryo, actually plots out in terms of wing loading together with the adults. So there's strong evidence here that this individual, even though it's still inside the egg, had the ability at least to fly. And this is in very sharp contrast to hatchling birds or baby bats. If you do the same thing, you find that generally speaking, their flight ability is 0, with the exception of megapodes. Ask me about those later. So what we can do in terms of wrapping up this exploration of the evidence for superprecocial flight ability in pterosaurs is have a look at 5 different criteria here: ossification, morphology, wing membranes, aerodynamic ability, and taphonomy. I've not spoken about all those. We've got 14 different pterosaurs in which we have embryos or hatchlings to look at. And when we do this, we find that in 60 out of 70 of these cells, the answer is yes, that's consistent with a flight ability. So in this particular case, I'm totally convinced that hatchling pterosaurs were able to fly. They certainly could fly if they want to. One corollary of that is it probably means they didn't need to have parental care. That doesn't mean to say they didn't get parental care. They might have still got it, whether they wanted it or not, but it doesn't mean that they required it. So, what does this tell us overall about pterosaur reproductive biology? They almost certainly had paired ovaries. They had relatively small eggs, which suggests, relatively speaking, a low parental investment. Clutch size was certainly, we think, greater than 1 or 2, but exactly how many we don't know. Hopefully there'll be data on this in the near future. They have parchment eggshells, which suggests incubation by burial, which suggests development at ambient temperatures and relatively slow development of embryos. We have a superprecocial flight ability, which is at least consistent with little or no parental care. We'll probably never know for sure, although who knows, trackways, you never know what you're going to find there. If we put all this together, what it tells us is that pterosaurs basically retain that basal sauropterid, or what you might call reptilian mode of reproductive biology. But to put it back in here, what it means is all the features that we see or that we can deduce about pterosaur reproductive biology are consistent with these kinds of extant taxa, in particular lepidosaurs. They don't actually share any of the derived features that are found in birds, which is interesting because typically pterosaurs fall out around about here in cladistic analysis. Richard's looming, I'll wrap up. So one of the conclusions, perhaps the most interesting conclusion we can reach, is that in this respect, pterosaurs are far less convergent on birds and bats than currently thought. And indeed, there are other aspects of their biology where we also are increasingly thinking that actually they're not like birds and bats at all. So the kind of takeaway message is pterosaurs, perhaps even stranger and more distinct from living flyers than we thought was the case previously. Thank you very much.
Dave. We filled up your entire 15 minutes, so we're going to have to move on.
Was my plan.
So our next speaker is Steve Fossati and the House of Thousands, talking about Scotland's Jurassic Park.
There you go, Steve. Here you go.
All right. Okay, thank you, Richard. Thank you, Phil. So I moved to Scotland almost 5 years ago now, and so what I want to talk about today is some of the work I've been doing since that time on the Isle of Skye, which is one of the best places in the world for Middle Jurassic vertebrate fossils. And this is work that's been done with a really fantastic team of colleagues and of students, this group that we call the PalAlba group, which has brought together a lot of the major institutions in Scotland to work collaboratively to discover and to study and to conserve Scotland's fossil vertebrates. A lot of the work we've been doing is focused on Skye off the west coast of Scotland, and for those of you that have been there, I'm sure you know Skye as this beautiful, enchanting place, these Tolkien-esque landscapes, and of course, some really crap weather a lot of the time. But regardless of the rain and the wind and the tides, we keep going back because Skye is really important. And that's because along much of the coast of Skye, the tidal platforms, the cliffs, we can find rocks of the Great Estuarine Group. And this is a series of sedimentary units that were formed in rivers and in deltas and in lagoons, beaches, nearshore marine settings, during the middle part of the Jurassic. And that's why these rocks are so important, because, for instance, when it comes to dinosaurs, we know quite a bit about the earliest dinosaurs from the Triassic and the early Jurassic. We know quite a bit about the iconic movie star dinosaurs of the late Jurassic and the Cretaceous, but we don't know so much about those in between, in the Middle Jurassic, just because there are not that many fossils of this age from anywhere in the world. And that's really frustrating because it seems like this was an interesting, critical period in dinosaur evolution, when the first birds were taking to the skies, when the first tyrannosaurs, the first really colossal long-necked sauropods were evolving. And this is true, this pattern of many other vertebrate groups. We just don't have many fossils from that Middle Jurassic, and Skye is one of the only windows into this time from anywhere in the world. And it's not like we were the first people to recognize this. People have been working on Skye looking for vertebrate fossils for many decades, going back to Bob Savage and his teams from Bristol in the 70s. A decade or so ago, Susan Evans, Paul Barrett from here in London led teams to Skye. More recently, Roger Benson from Oxford has been leading teams. And for the past couple of decades or so, Neil Clark from the Hunterian in Glasgow, over in the corner, has been doing quite a lot of work on Skye and has described and discovered lots of important dinosaur track sites and some of the first dinosaur bones from Scotland. Much of Neil's work has been in collaboration with a really remarkable local character on Skye, Dougie Ross, who grew up on the island and built the Staffin Museum. Literally built it from the ruins of a one-room schoolhouse. And he's the only person that I know who has built his own museum with his own two hands. And that's one of the reasons why last year PalAS awarded Dougie with the Mary Anning Award. And unfortunately, he couldn't come to Lyon to get it, so a lot of you probably haven't had the chance to meet him. But earlier this year, Neil and Tom Challens and I were able to present him his award when we went up to Skye for fieldwork. So he's very chuffed to get the award. So Tom, who's also in the corner over there, Tom and I started going to Skye together about 5 years ago. And recently our work has ramped up as this PalAlba team has come together and as we've gotten some National Geographic funding with collaboration with Roger and his team to do more work on Skye, to build up our field team. So what I want to talk about today are some of the newer discoveries and some of the newer studies that we've been doing. And we'll start off on the far northern tip of the island at a place called Duntullum, in the shadow of the ruins of a 14th century castle. This is what it looks like on a nice day. And if you go down to the beach and you wait for low tide, that beach turns into a big tidal platform. And so a few years ago, Tom and I took a small group of students there to do what Tom likes to do, look for tiny little fish bones and fish teeth. And we spent all day looking for these things on our hands and knees. And then as we were walking back to Tom's tricked out van. Later in the day, we started to notice that there were some depressions on that platform. And as our eyes got attuned to these things, we realized that there were actually quite a lot of these across the platform, and they seemed to form a pattern, kind of a left-right zigzag type of pattern. They had sediment deformation below them, so that means they must have been made when before these rocks were lithified. Some of them were filled, and there were casts that were raised up, and some of these casts had little projections on one side. Others of these casts were paired. There was a small one with a big one, and some of these casts were really quite huge, so up to 70 centimeters or so in diameter. So it dawned on Tom and me that what we were looking at were trackways, were handprints and footprints, and there's really only one thing in the Middle Jurassic that was walking on all fours that was this big, and that's sauropod dinosaurs. So it would have been some type of dinosaur that more or less looked like this that made these tracks. Probably about 10 or 15 meters long, 10 tons or so roughly. Now this image is made by a really excellent paleo artist north of the border named John Hode. And we've worked quite a bit with John to reconstruct what sky would have been like 170 million years ago when these sauropods were leaving these tracks. And so this is the image that John has come up with, this kind of dystopian post-apocalyptic scene of a few sauropods picking for plants in these lagoons. Of course, always a bit of artistic license with these things, but a lot of this is based on what we see in the rock. So there is evidence for really powerful storms in the Great Estuarine Group, and also we can tell that these sauropods were making these tracks in a lagoon. Their tracks are found in lagoonal rock, so they would have been wading in shallow water when they made these tracks. So Tom and I, we published a very short description of this site to start the conservation process going. But more recently, we've been using drones to take a lot of aerial images of this site to make high-resolution 3D photogrammetric models. And this is work that has been mostly done by our amazing master's student in Edinburgh, Paige DiPaolo. And so Paige has made this really detailed 3D model. Here's just a quick snapshot of that. What this model does is it helps make some order out of the chaos of all of these tracks on this tidal platform. And so what Paige has been able to see is that there's over 150 individual tracks at the site, and these form several trackways that cross each other, and they seem to generally indicate what we would call milling behavior, these sauropods just kind of hanging around in this lagoon. So meanwhile, while we've been working on a detailed study of this site, we've also been exploring other places in Skye for other track sites, and we've been finding new ones. And so let's go a little bit farther down the East Coast to a place called Brothers Point now, and this is what it looks like. Like Duntullum, at low tide, a lot of the surrounding area turns into a tidal platform, and like at Duntullum, there's quite a lot of dinosaur tracks on different parts of this tidal platform. And we have 3 new track sites from Brothers Point, which very imaginatively we call Brothers Point 1, 2, and 3. And so I'll very quickly say a few things about each of those sites. So we can look first at what we call Brothers Point 1, and this is a mudflat, an ancient mudflat. We have tracks, 3-toed tracks of theropod dinosaurs, similar to ones that Neil has been finding for the last few decades, but also a new type of track for Skye, smaller quadrupedal tracks that can be assigned to an ichnotaxon called Deltapodus. And this is widely considered to have been made by a stegosaur, so one of those very familiar plate-backed dinosaurs. And this is interesting because these tracks are probably a little bit younger than the oldest Deltapodus tracks, which are from Yorkshire, but they're probably a little bit older than the first stegosaur bones in the fossil record. So they're some of the first records of this very well-known group of dinosaurs. So that's Brothers Point 1. If we move over to Brothers Point 2, this is a lagoonal site again. We have more sauropod tracks. Some of those have really nice definition of the toes. And then if we move over to Brothers Point 3, we have another mudflat now. This was a site, by the way, that was found by a student of ours, Paulo, who was visiting from Brazil. and was utterly confused by the Scottish weather, but somehow on the rainiest and coldest day of the trip, he found this site. And so there's other theropod tracks at this site, but there's another new type of trackway for Skye. We have these big tridactyl tracks, three-toed tracks, only footprints, so this was an animal just walking on his hind legs. And these are different from the familiar theropod tridactyl tracks. The toes are fatter, they're blunter, they diverge from each other at wider angles, and these are features that generally correspond to what are often called ornithopod dinosaur tracks in the literature. So tracks that were made by plant-eating precursors of the duck-billed dinosaurs. And so the animal that made these tracks was probably about 5 or 6 meters long. And the best skeletons, the first good skeletons of those types of ornithopods turn up in the late Jurassic, but there's some tantalizing bones from the middle Jurassic of big ornithopods, so I think these tracks add to that picture. And so with these new track sites, with the sites Neil and Doogie and others have found, we now have a lot of data, and we can start to analyze those statistically to look for patterns. And what we see when we do that is a dichotomy. We see that the sauropod tracks are preferentially found in these lagoonal rocks, and all the other tracks, the theropods, the stegosaurs, the ornithopods, those are preferentially found in the mudflats. And so we hypothesize that this has something to do with environmental preference. So environmental partitioning, and this is something we can continue to test as we find and we describe more track sites on Skye. So that's a little bit about the dinosaurs, but as I always have to remind myself, it's not only the dinosaurs, there's a lot more. Dinosaurs are just one part of this amazing fauna from Skye. And so in the last few minutes I have, I'll just say a few words about some of the other things that would have shared this environment with these dinosaurs. So we can go a little bit north from Brothers Point, up the cliff to a place called Valtos. Big sheer cliffs, rock falls there. We can look for fossils in these rocks, very hard rocks, so we need to saw these things to get the fossils out, but there's sometimes some very delicate little surprises in there, like this tiny little bone that Tom found a few years ago that turns out to be the jawbone of a small crocodilomorph. So an early relative of modern crocodiles. And it turns out that there's another jawbone of a small croc from Duntullum, not too far from where those sauropod tracks are found. But it's a different type of jawbone, so we have at least 2 species. But more or less, they probably would have looked pretty similar. These were small, dog-sized, primitive crocs that were living in these lagoons, probably at the interface of the land and the water. Meanwhile, farther offshore, there were other types of reptiles that were ruling that ecosystem. And we can find some of their fossils a little bit south on the east coast at places called Rigg and Berrig Bay. And this is Berrig Bay here. This is where the Storlocks power station is. And in the 1950s, this guy here, Brian Shawcross, was walking around there. He found these 4 bones. He donated them to the Huntarian, where Neil has kept them safe for a while. And then a few years ago, as the PalAlba team came together, we studied these and identified them as a new species of ichthyosaur, which we called Yarkvara. Doesn't look like it's pronounced Yarkvara, but it's a Gallic name. And so, interesting discovery, but only 4 bones. Not so much you can do with 4 bones. But believe it or not, a few years after Brian made his discovery, another local, Nori Gillis, who ran the Storlocks power station, was walking along the beach and made a much more complete discovery. And he wrote about it to the National Museum of Scotland. They came up and collected it. They kept it safe for a few decades. And then recently, we've been able to get it prepared, and we've been able to put it on exhibit, and we're studying it now. So this is another ichthyosaur, but a much nicer one. There's over 100 bones here, so this is one of the more complete skeletons of a Mesozoic vertebrate from Scotland. And finally, just before I close, just a few words about some other things. So I won't say much about the small-bodied animals. This is the work that Roger and his colleagues are doing a lot of work on, and with Susan Evans and others. But my PhD student Elsa in Edinburgh is describing this incredible small mammal skeleton that was found a few decades ago, so stay tuned for that. And we also are starting to get some records of the things that were living and moving around in the air. So some of the things like what Dave was just talking about. So our most recent discovery last year, when we were out on Skye, was made by Amelia. Some of you may know Amelia. She just finished her PhD in Edinburgh with Rachel Wood, studying some of the oldest animal skeletons in the fossil record. But to take a break from Cloudina and Namacalathus, she came up with us to Skye, and she found this, which is the head of a pterosaur, which is complete from the tip of the snout to the back of the brain case. We can tell from the teeth it's a rhamphorhynchoid grade pterosaur. And as we started to take this out, we saw that it connected to a lot of other bones, so it looks like we have a pretty good skeleton of a pterosaur. It's with Nigel Larkin, one of the best preparators right now, so stay tuned. Hopefully we'll have something to say about that soon. And just in general, I would say stay tuned to Sky, because as the PalAlba teams come together, as we've been training a lot of great students, I think we're just scratching the surface, and there's a lot more to be discovered up in Scotland to reveal more about this Middle Jurassic ecosystem. Thank you very much.
Thank you, Steve. We have to move on to the next speaker. 15 minutes. Okay, so our next speaker is Daniel Mancher. Apologies for the pronunciation. He's going to speak about the pliable nature of the phylogenetic relationships within early ornithopods.
Okay, so. All right, so good morning. My name is Daniel Mancher. I'm from the Institute of Paleobiology of the Polish Academy of Sciences, and the topic of my presentation is the somewhat pliable nature of the phylogenetic relationships within early ornithopods. This presentation is going to be mostly based on a paper published a few months ago by me, by Clint Boyd, and by Martin Mazuch. Actually, you won't hear much more than you actually could see in the paper, because it's mostly based on this, and the topic of the paper was to describe a new basal ornithopod from the Czech Republic. Steve was talking about 4 bones, so I will talk about 1 bone. This dinosaur was first described in 2005 by a team of Oldřich Pfeiffer from the Charles University. It was first classified as an indeterminate iguanodontian, but it seems much more primitive, actually. The dinosaur is from Upper Cenomanian. It's about 3 to 4 meters long. Okay, it's just 1 bone, but we can infer this based on the size of the femur. And it was named after Zdeněk Burian and Josef Augusta. Zdeněk Burian is a well-known painter. He was a well-known painter. And Josef Augusta was a paleontologist who actually discovered Burian, so it seemed a good idea to name the dinosaur based on them. All right. While we try to infer the phylogenetic relationships or the phylogenetic placement of the new dinosaur, we face the same problem that many other scientists do while trying to deal with these dinosaurs.
Yeah, okay. Maybe.
Okay, so, but can you hear me? Yeah? Yes. Okay. Okay, so our question was actually, what do we actually know about these dinosaurs? Because they are, well, there are some problems with them. There is some, well, we can call it traditional contents of ornithopoda, but speaking of traditional contents is quite tricky because the phylogenetic hypotheses are changing constantly. So there are a few groups that were inferred as ornithopods during the last decades, but this is of course changing, like in the case of Heterodontosaurus that are now firmly placed near the base of Ornithischia. There is something like Thescelosaurus-like ornithopods that we can call Thescelosaurids. There are Hypsilophodon taxa, and then there are the traditional, the typical ornithopods that are called iguanodontians. All right, so I will now move to some groups of these, and I will discuss what we actually know about them, or if we do know anything actually at all. Basically, if it comes to ornithopods, they were divided into 2, let's call more or less informal groups. One is Hypsilophodon taxa, they were more primitive, and the other group was Iguanodontian taxa. Among those, the first one, the former, the Hypsilophodon taxa, we had taxa similar to Tesselosaurus from the Upper Cretaceous of North America, but they were not always inferred as a clade. There was, like, let's say, basal ornithopods. Some recent papers, such as that by Boyd and colleagues from 2009, inferred a monophyletic Tesselodontosaurid that consisted just of 4 taxa, and similar topology, which was a bit more species-rich, was inferred by Brown and colleagues in 2013. But these two studies actually did not study the position of these taxa on the Ornithischian phylogenetic tree. So the first one, to my knowledge at least, that actually studied the most abundant group of these Tesselosaurus-like taxa phylogenetically was Boyd in 2015. His paper was published in PRJ, and he actually inferred a relatively species-rich Parksosauridae. He called it Parksosauridae, but he now moved to the more popular Tesselosauridae. He actually inferred them outside ornithopods as a sister taxon to Sauropoda, to the large clade that's formed by Ceratopsians, Pachycephalosaurs, and ornithopods. So that was actually the first paper that suggested something like this. And then we have something very strange that's called Elasmarians. The name Elasmaria was first introduced in 2007 for just 2 taxa from the mid-Cretaceous to late Cretaceous of Argentina. These taxa were named based on their thin plates on their thoracic region, which is a very problematic character because later analysis they reconstructed different synapomorphisms of them, but I won't talk about details. It would be very boring here. But anyway, it seems that Elasmarians might be a thing because Boyd also inferred, they were basically first named just based on 2 taxa, Macrogryphosaurus and Telencoen. Boyd inferred them as a clade again. Basically, when they appear in a phylogenetic analysis, they always form a clade, so they really might be closely connected. Boyd also inferred another taxon, Notohypsilophodon, as their close relative. A later paper by Rosadia and colleagues in 2016 actually reconstructed a much more diverse Elasmaria, but they were pretty unresolved. So if they are a clade, we actually don't know, what was their internal evolution. In order to assess the phylogenetic placement of our new dinosaur, we used the data set first assembled by Boyd in 2015, or published by Boyd in 2015. We added some new taxa to this data set, which was basically expected to be used to assess the phylogenetic relationships of the whole Ornithischian evolutionary tree. We added some new taxa from different parts of the tree, including some Tesselosaurids, including some Dryosaurids, including some, there are even some Heterodontosaurids, some ornithopods. We significantly modified some taxa, such as we combined Lesothosaurus and Stormbergia based on study by Barron and colleagues. And we also modified Leninosaura just to reflect only the holotype, because a study from 2016 suggested that material from the type locality cannot be confidently assigned to the taxon except for the holotype. And we also updated some other taxa. Given that our analysis was based on that by Boyd, some results were pretty similar, such as Tesselosaurids were reconstructed outside Ornithopoda. But interestingly, or maybe that could be expected, Elasmarians were not part of this clade. Our analysis inferred Elasmarians as a basal and relatively species-rich clade in Ornithopoda, so it supports the conclusions, the contents of our elastomeric is almost identical to that of Rosadi and colleagues, so it supports their conclusions. What might be interesting, Hypsilophodon is also inferred outside ornithopods as a cystotoxin to Sauropoda, which, let's say, continues the trend to move those former Hypsilophodons outside Ornithopoda, and basically Ornithopoda is today almost identical to what was previously the Iguanodontia. And the basalmost ornithopod, according to our study, is Gideon mantelia. All right, but there are still some problems. And the most, apparent problem is the long Gauss lineages. For example, if it comes to this tree, which is from Boyd, his time-calibrated tree shows very well that if such topology is correct, then we have a huge Gauss lineage leading to Thessalsaurids. So if Thessalsaurids are really outside ornithopods and in this position, then they had to appear somewhere deeply in the Jurassic. And we have no Jurassic taxa belonging to this clade. So what can we do in future to actually, change this or make it more accurate? Ornithopods are not studied using more than a single phylogenetic method. So it might be a really good idea to use other research strategies or use entirely different phylogenetic methods, such as Bayesian inference. All right, I would love to thank people that made this actually possible, and especially Boris Ecker from the National Museum in Prague, who granted me access to the type specimen. And I would also love to thank Edyta Felsin from Poland, an extremely talented illustrator who made this extremely improbable restoration of Buryanosaurus. So here are the references that I used, and I thank you very much for your attention.
Thank you. We've got plenty of time for questions. If anybody's got anything. We've got a couple of minutes if there's no questions, so we'll just wait a second, make sure we keep on time. Yeah. Let's talk as much.
So thanks for coming back. So we have a session of a variety of talks which run from now until 12:45. So we start with Alexander Askew speaking on behalf of himself and Charles Wellman on the Middle Devonian Catch-Up event, its identification and effects in northern Spain.
Thanks, Michael. Right, so yes, this is my work on the Middle Devonian Catch-Up extinction event and what it's been doing in what it did in northern Spain. Feel free to tweet this and what have you. I've no restrictions on that. Right, so we know that the Devonian period is a very interesting one in the history of life. The marine communities are becoming very complex. There's lots of fish knocking about. And we also have the very first forests developing on land. These are strange-looking forests full of strange plants, but it was into such forests as these that early tetrapods emerged in the Upper Devonian, really. We also know that the Devonian period is punctuated by a large number of extinction events. Here they all are in this column here. These are pronounced in the marine realm, and they tend to be associated with marine transgressions leading to some degree of benthic anoxia and corresponding extinctions in benthic fauna. The one I'm interested in is this one, the Catch-Up event. This is taking place on the Iphelian-Juvetian boundary in the Middle Devonian, roughly anyway. And it's marked by thick black shale horizons in Central Europe. It's known in the Americas, Morocco. I think it's known in China as well. But there's other pretty big parts of the world where we don't see these shale horizons particularly prominently. And in keeping with a lot of these other events, its origin is a mystery. We don't really know why it happened with any degree of certainty. So I've been looking for it in northern Spain. Now, why go to Spain, you might ask? Well, it's got lovely scenery, but it also has a really interesting setting in the Middle Devonian. It's recently broken away from northern Gondwana. It's in this chain of isolated land masses on the southern edge of the Rheic Ocean. And these are surrounded by a shallow sea, into which was deposited a complete record of sedimentary sequences through the whole of the Devonian period. As I said, this is a shallow marine environment, and it shows a really odd pattern of alternation between calcareous and siliciclastic deposits. This is weird because these two rocks are deposited in completely different settings from each other. Something quite major needs to have happened on each one of these transitions in order for this pattern to have emerged, and for it to have persisted over such a long period of time is quite odd indeed. I've been looking at the Naranco, Huergas and Gustala Piedra formations of Asturias, Leon and Palencia provinces. I should point out these are one formation. It just has three different names because of the three different people who studied it. And they represent this predominantly sandstone interval from the lower Iphelian into the lower Juvetian. This straddles where we should find the Catch-Up event, and it's, as I said, it's a predominantly sandstone formation, but you also get intercalations of silt and mudstones in there as well that can be quite rich in organics. This is where the Middle Devonian rocks outcrop in northern Spain. They start up on the northern coast near Gijon, where the sediment is very sandy. There's a very strong terrestrial influence. That influence weakens as you move around this arc shape, south through Asturias, into Leon, and then east towards Palencia. By the time you're in Palencia, you're basically looking at marine limestone, but you still get those finer beds of silt and mud interspersed throughout the whole shape. So we clearly have a contiguous formation, and there's probably a terrestrial source off to the north somewhere. That seems to be where the sediment is coming from. This is what that looks like geologically. You can see it's an extraordinarily complex picture. It's very, very heavily folded. This is all caused by the Cantabrian Mountains that are sort of here and more off to the east. The Devonian rocks are picked out in, it's meant to be dark green, it's sort of brownish, around this shape here. You can see they're extremely heavily folded by the tectonic activity, but they're quite well exposed. And all these black crosses show where my sites are. These are all outcrops of one kind or another, road cuts, sea cliffs, that kind of thing. And these 4 arrows show significant sections through the formation which I've logged and sampled. Now that gives me a good stratigraphic control on my samples. My methods are an entirely ordinary palaeontological maceration. Take your rock, stick it in acid for a bit, HCl, HF. That gets rid of most of the mineral content. And then sieve out what survives, heavy liquid centrifugation to get rid of clay, and then all my material is thermally mature to some extent. It's all been heated up during burial, and so I've oxidised all of it with Schultz solution, and that lightens up the colour and makes it a lot easier to look at under a light microscope. Once I've done all of that, I've then found a really rich assemblage of palaeonomorphs of all different types. So these ones are acritarchs, these are marine algae essentially. These 2 are chitinozoans. We're not really sure what these are. I've been known in the past to describe them as enigmatic thingamajigs. These are, again, marine forms. We can definitely tell that. Some benthic, some pelagic, we're not really sure what their biological origin is. Skelecodonts are marine as well. This is the tooth of a polychaete worm, quite rare in this assemblage, but they're definitely here. And then the terrestrial flora is represented by these spores. These are the reproductive particles of land plants, and they come in all different shapes and sizes. And this reflects the complexity of those forest communities that I mentioned earlier that have just appeared. Now, these groups have a lot of uses. Their proportions can tell us about the environment. If I've got lots of spores, then the coastline must be close by, because these have to disperse from the land. Conversely, if I've got lots of acritarchs, I'm probably further offshore, because these come from the seas. On top of that, certain species are known to occur at certain points in time, so they have a biostratigraphic use. I can date the formation, hopefully with some accuracy, and that's what I'm going to take you through now. So this is a lithological column of the Naranco formation that I made at the Playa del Tranqueru site. This is my best site because it preserves pretty much the whole thickness through the formation. You can see the bottom, more than half of it, is sandstone. This is expected, this has been seen in previous work. The top half of it is very heavily interbedded with finer beds of silt and mudstones. This is handy because it's this lithology that preserves the palaeonomorphs, but it also indicates an environmental change is happening. The fine sediment is deposited in deeper water than the relatively coarse sandstone. And so what we have here is cyclical sea level change. The water levels going up and down across whatever time period this might represent. In order to date rocks of this age, one of the best ways to do it is with biostratigraphy, with their fossil content, and conodonts are one of the best things for that. Their zonation is very well chronologically controlled. Trouble is, you don't get them in the Naranco. It's the wrong kind of lithology to preserve them. But you do get them in the Candace and Moniello limestones above and below the formation. So we're able to, unusually at this age, we're able to bracket the age of this sandstone quite accurately. So the base of it is no older than the lower Polygnathus costatus conodont zone, and the top of it is no younger than the lower P. varus zone. So we are looking at lower Iphelian into lower Givetian, as I showed previously on that stratigraphic scheme. So where do my fossils come in? Well, this is Geminospora lemurata. This is an important spore. It's probably of progymnospermous origin, and it defines its own biozone in a lot of different schemes and has a very well-defined point at which it first occurs in the lower, but not lowest, Givetian in the P. hemianzatus zone just here. So any sample that contains this spore cannot be older than this point. I find it just here, in only my third sample that I have at the Playa, and it also occurs at the base of my other two sample sections as well. So I can say with some confidence that this heavily interbedded part with all this sea level change must have been deposited within this relatively short space of time between the inception of G. lemurata and this upper conodont point here. This indicates that not only do we have cyclical sea level change, we also have rapid deposition. To get this thickness of sediment in what is a relatively short space of time, we must have some kind of increase in the rate of sedimentation. This isn't like normal, really. Now, remember the Kachak event? Well, this is taking place just here on the Polygnathus enzensis zone, from slightly below, slightly above. It ends before the inception of G. lemurata. Now, elsewhere in the world, the Kachak is marked by black shale horizons. I thought that this interbedding, this darker organic-rich sediment, might represent the Kachak here. Clearly, it doesn't. It's too young. It is entirely after the event. So at best, we may have the tail end of it, probably not even that. It has been previously hypothesised by John Marshall and others looking at the Orcadian Basin that the Kachak was caused by an increase in insulation, which leads to eustatic sea level rise, and a greater monsoonal climate, leading to greater terrestrial runoff, and all of this increases stratification and increases the benthic anoxia that we see elsewhere. This would also give you an awful lot of nearshore sedimentation, you would think, if you've got all this new rainfall washing stuff off the land. We do not have that here. We have rapid sedimentation, and I should point out I also have sedimentological evidence of this. It's not just the biostratigraphy I'm going on. So it's not represented here, but we still have this great big sandblock study doing nothing. What if this is the Kachak event? Well, I think it's not outside the realms of possibility. Elsewhere, it's marked by black shale horizons, but don't forget, this is a really near-shore environment. I don't think this is deep enough to really generate the kind of anoxia that you would need to produce a black shale, or a proper black shale anyway. Maybe a really great thickness of rapidly deposited sediment is a signal for the Kachak in a near-shore environment. I think this idea has some weight behind it. I have no sample sections from this sandstone, so I can't date this and I can't say this with certainty. But I definitely think that in a near-shore habitat, a great thickness of rapidly deposited sediment is a good signal to look at for if you're looking for the Kachak event and you can't find any of that black shale. So, where does all this leave us? Well, I have a really rich Middle Devonian assemblage in northern Spain, far richer than has been previously described for the area. The biostratigraphy and the sedimentology supports the idea that this formation was deposited really rapidly, and it's probably discontinuous as well. It's got gaps in it just by the nature of the sediment and how it gets deposited. And I think all of this supports that monsoonal hypothesis for the Kachak event, and I think that rapid deposition is a good thing to look for if you're trying to find the Kachak event in a near-shore environment, as well as the fact that I also have this, I think, support for this hypothesis in a completely new continent from Scotland, and we're moving towards an idea of this, a better idea of the Kachak as a truly global event. With that, I will leave you with an appropriately wintry shot of the Embalse de los Barrios de Luna, and thank you all for listening.
Great, thank you. There's time for a question or two, if anybody has one. Yes, John.
Yes. Where does all that sand come from?
Well, I can only say the nearby land. The Playa del Tranquero is definitely near shore. There's a lot of spores in there. I can only say that it's from the nearby land.
So are you worried about the size of that land compared to the volume of sand?
Not really, because there's not really any extant evidence for it. It's off in the sea somewhere. I can find little work on it beyond maybe it's coming from the north. You know, it seems like not many people have really thought about what it looked like, how it was.
Any way that you can go and check other sections that will show you like more offshore environments, so where you can compare your record?
I could do that. The problem is that as you get closer to Palenthea and further offshore, the samples get worse and worse and worse. So my Palenthea samples are really shocking. With comprehensive sampling, I think I probably could. And that's something that I wouldn't mind doing in the future, as well as possibly looking at this in other parts of the world or slightly different time periods or something, to look at these other extinction horizons that we've got. So that's a definitely good idea.
Okay, thank you very much.
All right, thank you. There we go.
Somewhere near to back there, it'll be fine. Okay. Hello, can you hear me?
No, it's not micro, it's just recording.
Oh, right, okay, right.
Just be loud enough for the room, that's all.
Okay, fine.
Okay, thank you. So next is Max Stockdale from Bristol, who will be talking about climate change and rates of crocodile morph body size.
Thank you, Mike. I'm going to open up with a bit of an apology because I have a cold and my voice is not behaving the way I would normally like it to. So if anybody needs me to repeat anything, please say so. Yes, so as Mike said, I'm going to be talking about climate change and rates of crocodile morph body size evolution. Climate change obviously is an extremely important and concerning issue for everybody, and crocodilomorphs, it would seem, are the perfect group for studying its effects on large-scale evolutionary processes. Before I start, I've got some thank yous to hand out, most of all to Mike for supervising the project and putting up with me and my crazy ideas, to Emily and Graham for all their fantastic advice and suggestions on my thesis, and to Armin, Tom and Mark for teaching me some of the methods. Also, last but not least, to NERC for funding the project, which is very nice if you can get it. So the crocodilomorphs, on this slide there is a picture of every crocodilomorph in existence today, give or take one or two. There's a debate about, which is it, this chap here, the dwarf crocodile. Some people think it might be two species, others think it's just one. So there's about 24, 25, give or take, in total. On there, it might look like a lot, but in the grand scheme of things, that's really not very much, because the common ancestor of all modern crocodiles first appears in the Cretaceous period, but the crown group Archosaurs, so crocodiles and birds, first appears in the Triassic, and the sister group, the birds, have achieved a diversity of over 10,000 species in the same amount of time. So crocodiles, they're really a bit pathetic, really. There's just not very many of them. And the diversity is also not very high either, the morphological diversity, that is. They're all aquatic, they all look kind of basically the same, they're all amphibious ambush predators, with long noses and long tails. So the diversity is quite low, and that's sort of, why is that? That's really quite weird. Actually, the geographic distribution is also quite limited. They're constrained to a band about 30 degrees north and south of the equator, and that seems to be related to temperature, because of course they're ectotherms, they have no control over their body temperature. So they're limited to regions of the planet which are warm enough to sustain them. That's in stark contrast to what you see in the fossil record. Yes, we do find in the fossil record sort of familiar things like this in the top right. Crown group crocodiles, as I say, they appear in the Cretaceous period. But there's other types of crocodilomorph, I should say, in the Mesozoic, which are a lot less familiar. We have thalattosuchians like this one, highly derived ocean-going crocodiles with fins and flippers. And then we have these strange, sort of almost mammal-like notosuchians, heterodont. Some of them might have been herbivores, quite alien creatures by comparison. And we also see cursorial forms like these two at the bottom. This is a rather primitive one from the Triassic. But high-walking, hypercarnivorous crocodilomorphs appear in the fossil record of South America as recently as the Eocene. So we've got this much broader spread of diversity. But crocodiles, they have this blessing. A lot of them live in water, and many of them have tough body parts like reinforced skulls and osteoderms. So as fossil vertebrates go, their fossil record is actually pretty good, which makes them excellent subjects for studying large-scale evolutionary processes. So some of you might remember this painting that appeared in the news earlier in the year. This is Machimasaurus rex. And one of its outstanding features is that it's pretty big. It's larger than any crocodile you see today. And this is an ongoing theme you see in the fossil record, that body size is much more variable in fossil crocodiles than it is today. So we have this enormous thalattosuchian from the Middle Jurassic period over here, and then, by contrast, we have this basal one, this Terrestrisuchus found in Gloucestershire, which is tiny. It's only the size of a cat. So you see this enormous variation, much wider disparity in body size than you see today. body size is really important because it affects all manner of different aspects of ecology. Things like physiology, the rate at which it grows, the rate at which it can reproduce, its geographic range, the size of its population, all sorts of things. So it's quite an important question. Why is there so much more variation in the past than there is today? Body size is an attribute which can be ascribed to anything. It's not like certain characters which are present or absent. Everything has a body size, so it's an attribute you can measure for everything. And also, you don't need a complete specimen to measure body size. You just need a relative proxy, something conservative that doesn't change very much. And for the purposes of this study, I have assembled a data set of about 190 measurements of skull width. The skull width seems to be quite limited in how much variation it has with body size. Now, if we plot body size through time for crocodilomorphs, I've gone from the lowest Jurassic because that's where the fossil record starts being good enough you can actually do it. You can plot body size through time, and underneath here, these dots here, these are delta 18O values, so it's a proxy for temperature. It's a little bit counterintuitive because delta 18O is reversed, so the more negative it is, it's actually warmer. So these points correspond to cooling through the Cenozoic, so it's relatively low temperatures today, warmer in the Mesozoic, which is sort of what we all understood. But as you can see, there's actually quite a strong correlation between body size and temperature. And that might seem counterintuitive when we think of the giant crocodiles from the Mesozoic, things like Sarcosuchus and Deinosuchus, these enormous dinosaur-sized crocodiles. But actually, those giant crocodiles were sort of bucking the trend. And many of the largest crocodilomorphs in the fossil record are actually from sort of the Miocene onwards, things like Purosaurus, Gryposuchus, Euthecodon. Even the modern saltwater crocodile is one of the largest that's ever existed. That's this guy down here. So my question is, are we able to look at this in a more sophisticated way to see if this trend is actually real and sort of what's going on with it? So I wanted to look at this in a comparative phylogenetic context. Comparative phylogenetics allows us to estimate trait trajectories by reconstructing common ancestors and projecting character states through time. And it would be looking at it in terms of evolutionary rates, that is, phenotypic change per unit time. And to do this, you need to have a phylogenetic hypothesis of, in my case, the entire Crocodilomorpha. That's actually not a small task. So what I've done is assemble what's called an MRP supertree, MRP being an acronym for matrix representation parsimony. And it's the best method because others are simply not practical at this scale. Supermatrices accumulate huge numbers of inapplicable characters. For example, lots of matrices use characters from osteoderms of crocodiles, and lots of crocodilomorphs don't have osteoderms, so you just end up with these big blocks of question marks in a supermatrix. And they become so cumbersome that branch support is very low if you're using parsimony, and Bayesian analyses simply never converge. They just won't find a satisfactory answer. And also Bayesian supertree approaches, they're not particularly widely used, but there is a way of doing Bayesian supertrees. But in the same way, it takes a very, very long time to actually reach any kind of convergence or consensus on a tree. So MRP was the only practical way of doing this. Essentially, it works by taking published trees and then deconstructing them into a matrix of virtual characters. That is, so each node on a source tree becomes a character. So in this top tree here, there's a character which is common to alligators and crocodiles, and then those two taxa are encoded as the character being present on the matrix and then absent in all the others. And then you can reanalyze this matrix to produce a single tree. Now, I did this in TNT, and I've evaluated the relative effectiveness of each supertree using this equation. This is the Steele and Enrigo formula, and it essentially compares each of the winning supertrees with the source trees, and the tree which has the greatest in common with its own source trees is actually the best supertree, and that produces a phylogeny. Now, this is where it gets a little bit more interesting, that I've taken this phylogeny and the body size data I mentioned before, and I've brought them together into a phylogenetic model. I've used two different pieces of software for this. I've used Bayes traits and Geiger. There's a wider diversity of models established in Geiger at the moment, and actually at the time I was using an older version of Bayes traits which didn't have as many models in it. And the best fitting model turned out to be a variable rates model. So the rate of phenotypic change per unit time is variable. It's not consistent across the tree. And it produces this tree that I've put here. I don't know how, I hope you can all see it because it's the blue on the black background isn't working very well. But the branch lengths that you see there are actually, it's not time, that's actually evolutionary rate. I've rescaled the branches so that you can see which ones are evolving more quickly. The longer branches with the hot colours are evolving faster. The shorter branches with the cool colours are evolving more slowly. And you see this sort of pattern that the larger, more inclusive branches are evolving more slowly, and the faster, more rapidly evolving branches are limited to small clades and perhaps even to single taxa. And I'm interpreting this as a sign of kind of a punctuated equilibrium model of evolution, that we've got long periods of relative stasis and lack of activity that's being interrupted by these short bursts of violence. So it's almost like the evolution is static. It's not moving around very much, except periodic shifts, perhaps environmental changes, that are creating these sort of jumps in evolution that don't last very long. There's also considerable support for an Ornstein-Uhlenbeck model. This is where a phenotype is constrained about an optimum. The Ornstein-Uhlenbeck model presents a problem because it's very prone to false positives. I'm not saying it doesn't exist, it's just extremely difficult to distinguish from a Brownian motion model. So that support needs to be considered with a great deal of caution. But these rescaling values, so the numbers I've used to actually use from the model, which I've used to set these branch lengths, can actually also be plotted through time in the same way I did with body size. And that's what I've done here. This is the same thing where we've got this delta 18O values for temperature on the bottom, and then here is a smoothed curve of rates of body size evolution. And here's a simple regression analysis. Again, high rates of high correlation between rates of body size evolution and temperature. And again, it's positive. So increasing rates of evolution occur with increased delta 18O, that is increased cooling. So when it gets colder, rates of crocodile morph body size evolution goes up. And I think what's going on here reflects what Paul Markwick and colleagues found out in the 90s, that crocodilomorph physiology and evolution is constrained by temperature. In this case, they seem to be getting bigger when it gets cooler, and they think this is to do with body size surface area to volume ratios. That is, that the surface area is relatively smaller if they're bigger, and that helps them retain body heat as temperatures drop. And I see there's actually real cause for environmental concern about this. I mean, crocodiles are already becoming invasive species in certain parts of the world because as temperatures warm up, their habitable range is getting wider. Yes, as for example, crocodiles that are in farms, sometimes they escape, and now they're establishing themselves as invasive species in certain ecosystems. But also, if there's this connection with body size, which my work seems to be indicating, crocodile prey choice is linked with body size. They attack bigger things if they can tackle it, essentially. So the bottom line seems to be that if temperatures go up, then average crocodile body size might go down, and then the predation pressure they present in an ecosystem might shift, and that's going to have untold consequences to food webs where crocodiles are natively present. Just to wrap up, body size evolution in crocodile models seems to be fitting a variable rate model. Body size and rates of body size evolution increases with cooling. Higher evolutionary rate is limited to less inclusive clade, sort of a punctuated equilibrium court jester type of model. Yes, I think that about covers it. So thank you very much for listening. You've been a wonderful audience. Does anyone have any questions for me?
Just a point you said about the surface area volume ratio. Is it immediately apparent that an ectothermic animal that heats up by bathing in the sun will, because if its surface area raises relative to its body, it'll be able to absorb heat faster, which would be the opposite of what you think of an endotherm losing heat from its surface?
I see what you mean. So you're thinking that an animal would want to be smaller because it would have a relatively large surface area to obtain heat more quickly.
Yeah.
I see what you mean. My response to that would be, if the climate is naturally warm, they're not going to be getting opportunities to radiate that heat. So if it's the climate is stable, then it's less necessary for them to absorb heat through the surface in the first instance. So they don't need to warm up as often. But I do see your point. You need to give that some more thought.
One more question for Steve.
Steve?
Max,
Mm-hmm.
the baseline data collection for your body size proxies, have you considered species that are well represented by numerous specimens, considering these are animals that grow throughout their lifetime.
Exactly. You've hit on one of the problems with this sort of data. I've had to make the assumption that they're adults on the grounds that adulthood accounts for a greater percentage of its total lifespan. So it's unlikely that juveniles would be preserved. But also, like you say, it's going to be it's a problem because crocodile adulthood doesn't really have like a discrete start. They grow throughout their life. So this is like a measure of how big can they get. So it's sort of like, you know, they can at least get to that size, not necessarily any smaller, but the upper limit is not below that. But you're right, it's a caveat.
So next, Sylvia Denise and Stephen Holland on faunal response to sea level in the Jurassic Sundance Seaway in the United States.
Good morning, everybody. So, why do I want to use the stratigraphic paleobiology approach when I'm trying to understand the relationship between faunal and environmental change? So the reason is that whenever we want to ask this question, we have to consider that the distribution of fossils is not just controlled by the ecology and evolution, but very importantly by the stratigraphic processes that govern where fossiliferous sediments are deposited and preserved. So what if we can't have a measure of faunal change through time, through having a quantitative paleoecology, and if we can link it to sequence stratigraphy, so we can have a well-constrained temporal and environmental framework, then we can be more sure that the record that we are looking is really driven by ecology and evolution and is not just linked to the stratigraphic processes. And I believe that this approach can give us can give the opportunity to help answering important questions that we have in paleobiology. For instance, what drives the alternation of stability and turnover in ecosystem? So we know there have been many studies addressing these, that stabilities is promoted by strong ecological interactions among species, by habitat tracking, or wide geographic range and niche breadth, or also by population abundance. But what about turnover? We know that turnover happens in ecosystem like in a pulse, rapid way sometimes, or sometimes it's more prolonged. And usually it is linked to environmental perturbations, such as fluctuations in sea level and climatic shift. But then what is really driving this turnover? Because we know, for instance, that sea level cyclically change through time, but not all these changes trigger turnover. So what is the threshold effect? And I think that stratigraphic paleobiology can really help in better understand these questions. And specifically, what environmental perturbations trigger turnover? And when we found turnover, it is uniform along environmental gradients. So if we think about a marine environment across an onshore-offshore gradient. And so to answer this question, our contribution comes from the Jurassic Sundance Seaway. So why the Jurassic? Because it was a very important moment in the evolution of life that drove to the onset of the modern marine fauna. And why the Sundance Seaway? Because it was an epicontinental sea. And we know that epicontinental sea are fundamental because most of the record that we have from the Jurassic downward or backward in time come from epicontinental sea. So the better understanding of this system is very important. And the Jurassic Seaway developed in the North America from the middle to the late Jurassic, as you can see, had a very peculiar shape because it was very elongated and it had a single entrance. So if we want to address how fauna could colonize it, there was just this way that could enter. And it had an evolution that was 30 million years. This marine part of the seaway was 30 million years long. And as you can see, it was very elongated, and it was the seaway developed in a retroarc foreland basin, so we have the deeper part in the foredip here to the west, and then it became like shallower going through the craton side of it. Our study area encompasses most of Wyoming, and so why did we have such a big field area that, if you consider it's almost quite big as France? Well, because we wanted really to be able to see which were these onshore-offshore gradients in marine fauna, marine microinvertebrates through time. So we studied our section going from onshore section to offshore sections into the forest deep. So we studied 44 localities in 2 field seasons, and at each of our sections we did a lot of field work. So we measured stratigraphic sections, we interpret them in terms of sedimentology and sequence stratigraphic surfaces, and also we collected quantitative samples for microinvertebrate. And we built a data set that was mostly made by bivalves, both aragonetid and calcitic, but we also had brachiopods, crinoids, and belemnites among the most important taxa. And so through the sequence stratigraphy interpretation, we were able to subdivide our sedimentary succession into 8 third-order depositional sequences. That goes from J1 in the Bajocian to J5 in the Oxfordian. So these were, the depositional sequences were packages of rock that were bounded below and above by basins way unconformity, which were representing subaerial exposure. So we know that there were 8 main cyclic sea level changes across the seaway. And then through the sedimentological study, we also observed that in the seaway there were 2 main depositional systems: a carbonate rum system that was mainly developing in the Middle Jurassic, and a siliciclastic shelf which gradually passed into the Upper Jurassic. And so in our onshore-offshore study, we look at carbonate rump environments, and we sampled, here are our sample through space and time, and we sampled in the carbonate rump peritidal, huid shoal restricted and open shallow subtidal environment, deep subtidal and offshore. While in the siliciclastic shelf, we had samples from tidal channel environment, offshore transition, and offshore. So we put together this data set and we analyzed it using multivariate methods. So we had 184 sample, 120 taxa. So multivariate ordination methods are just a way to simplify the relationship, the multidimensional relationship that we have between multiple samples and multiple variables, which are our taxa. So we can have simplified that and see them in 2 dimensions, and this is here through a similarity matrix. So if these are 4 of our samples, the more they are similar in taxonomic composition, the more they will close in this space, in 2-dimensional space, and the more they are far away, the more they are different taxonomically, the more they will be far away. And so what are our results? So this is a multidimensional ordination, and what you observe, each of our point is one of our sample. And here I have labeled the sample according to their depositional sequence. As I said, they go from J1 to the Bajocian to J5 in the Oxfordian. And so you can really see that is a shift in taxonomic composition going from the middle to the Upper Jurassic, because they sample plot in different parts of the graph. Then we can label our sample according to the depositional environment. And really you can see that there is not just a change through time, but this is driven by the shift from carbonate to siliciclastic deposition. So we have this turnover. But what is most important and to observe is that when we label and we observe how our samples change if we label them according to the facies association where we have found in each of the 2 environments. And that's the important thing, because we observe that the samples are plotted according to an environmental gradient, onshore-offshore gradient, and that's very common in marine environment. But actually we found 2 gradients, because here in the center of the ordination we have the shallow water facies, both from the carbonate and the siliciclastic system, while as we go to the left side we have the deeper water facies from the carbonate system, and on the right, extreme right, we've got the deep water facies from the siliciclastic system. So what is it telling to us is that through time we had less turnover in shallow water, because you have that open shallow subtidal, peritidal, and huid shoal from the carbonate system are closer to offshore transition in tidal channel, so the shallow water samples from the siliciclastic system, but there was high turnover in deep water, because samples plot really far away from the other, the opposite part of the graph. So what is driving this pattern? So now we can do the same plot, but instead of plotting, oh, these are many species. Instead of plotting the samples, we can plot on the ordination the species that are driving this pattern. And here in red, I have highlighted the most common, the most abundant shallow water species. And what we observed that there are some species, like Laiostra strigilecula and Melagrunella curta, that they occur in the seaway from this opening to this closing. So from the Bajocian to the Oxfordian point sequence J1A to J5, for the 13 million years, they persisted inside of the seaway. So these species were not restricted to a single facies sequence or the positional environment, while the record in the deep water is controlled by turnover at the species level. We are different species of the same genus that have a third-order sea level cycle are colonized in the seaway. Here in orange, I put the Gryphea. It's a very common species in the Jurassic. We have Cravea planoconvesta in sequence J1A. And then we have in deep water Gryphea nebrascensis in J24, and then Gryphea sp in sequence J4. So we have in the deep sea stenotopic geological short-lived species. But again, how can we interpret this data? Why do we have this record? And we think that there is an important role driving this pattern, an important role in the variation of the area of marine habitat through subsequent sea level cycles. So here is a model showing eustatic sea levels, and this is how much variation you have in shallow water environment in the area that they have available to the marine species. And here you have the variation in area through multiple cycles in the deep sea. And you see that in the shallow water, you have much wider variations. What does it mean? It means that through time, you will have higher extinction in the shallow water environment, and this will lead to you will have a higher abundance of extinction-resistant species that will be eurotopic. There will be more abundance and they have a wider geographic distribution compared to the deep sea. So to conclude, what we observed is that there was turnover at the switch of the carbonate to siliciclastic deposition. So this was the main driver, but turnover was not uniform across the onshore-offshore gradient. But we have resilience in shallow water communities to third-order sea level changes and to the main change in the positional system. And instead, there was offshore turnover at the species level through time. And we think that its important role is the differential change in the area of marine habitat during sea level cycles, and this really can drive these ecological and evolutionary patterns. And I would like to thank all my collaborators and European Union. Thank you.
So we have time for a couple of short questions. Yes.
Do you have any evidence of invaders coming from outside the seaway?
Yeah, that's a good question. That we don't know if they were actually evolving inside of the seaway or if the species were coming from outside. And I think the main problem with this is that the taxonomy, as it has been developed over the years, is very local from the United States. I believe that many species are similar to the one that you would find in the Middle Jurassic of the Tethys, for instance. But at the moment, there is not a consensus on the taxonomy, so it's difficult to tell. But yeah, I believe that they were probably migrating, going up the Viking corridors and maybe entering in the seaway.
Anything else? Yes.
What about differences, for example, in species with different ecologies, so things in form metaphor, things in water columns, any differences that showed up?
Yeah, that's a good question. Actually, we have made like a biofascist study to divide them, but I haven't looked at that in particular. Yeah, so that's interesting. Thank you.
Okay, thank you. So the next talk is given by Catalina Pimiento on behalf of a number of co-authors on ecological consequences of extinctions from giant sharks to small mollusks.
Thank you, Mike. Thank you. Thanks. Well, today I'm going to be talking about two independent projects, but I'm going to try to combine them or connect them in this talk. First, I want to start by talking about habitat loss, because I found that surprising that habitat loss is the most important threat for biodiversity nowadays. And that's the same throughout geological time. So there is the sea level changes associated with habitat loss have been widely associated with extinction events through time. After the last mass extinction event and throughout the Cenozoic, the most dramatic changes in sea level occurred in the Pliocene and Pleistocene epochs. And even though these changes in sea level have been associated with some regional extinctions, for example, of some corals in the Caribbean, it has been widely stated that vertebrates remain resilient or resistant to these environmental changes. However, we know that there have been some examples of isolated extinctions during this time. For example, the extinction of the biggest shark that ever lived, that I'd worked in most of, Megalodon, became extinct around that time. Also some species of sea turtles, seabirds, and marine mammals. There was a significant decline in the diversity of cetaceans around that time. So our research asked the question whether these extinctions were isolated examples of extinction, of independent extinctions, or if they were part of an extinction event. So to answer this question, we assessed extinction rates through time and assessed if there were higher extinction rates around this period. The next question was whether these sea level changes cause these extinctions, or most specifically, if these changes in sea level cause habitat loss. And finally, our last question was about the effects or the ecological effects of these extinctions. Particularly, we wanted to know if there were any changes in functional diversity around this time. And this is where I'm going to connect the other project, because we're comparing now the effects of this extinction of marine megafauna, big animals, with those of small invertebrates in the Caribbean. So to answer the first question, we gathered a big data set from the entire Cenozoic in the paleobiology database, and we gathered all the occurrences, as opposed to first and last occurrence of each taxa. And we used a software called PyRate, Pi for Python and Rate for Rates, that Daniel Silvestre, one of our co-authors, developed. And with this software, we use all the occurrences, and we calculate times of origination and extinction at the same time as preservation probabilities and sampling probabilities, and with that, extinction rates. We found that during the Pliocene, there were higher extinction rates than during the rest of the Cenozoic. In fact, the Pliocene extinction rates were 3 times higher than the mean Cenozoic level. We did a better resolution kind of analysis, and we found that specifically at the end of the Pliocene is where these extinctions were higher, very close to the Pliocene-Pleistocene boundary. When we look at the proportional extinction, we found that each group of megafauna, I have to say that megafauna includes marine mammals, seabirds, marine turtles, sharks, and rays. When we combine them all, there was a proportional extinction of 36%. So around 1/3 of the taxa became extinct around this time. Now the next question was about changes in sea level and whether or not they resulted in habitat loss. So what we did is to use these sea level curves, and for each of these sea level scenarios, I think the time scale was 100,000 million years, we calculated the area available, the coastal area. So we calculated the bathymetry, and we selected the areas that were up to 200 meters, and we regarded that as coastal habitats, and we quantified that area. And this is what we found. So the Pliocene available area, the mean area, was much higher than the one in the Pleistocene. But when we look at the specific changes per each sea level scenario, we found that there was a dramatic drop in area at the end of the Pliocene. And after that, there were very violent fluctuations. We hypothesized, or we proposed, that these changes in sea level and in area or in habitat cause these extinctions. And this hypothesis was reinforced by some trait-based analysis we did. I'm going to explain this trait-based analysis later in the next slide, but for now, just know that we assigned these traits to each genera. And we performed some generalized linear models, and we found that thermoregulation was the trait that better predicted this extinction, with animals that were mesotherms and endotherms, so those that are able to control their body temperatures, at least at some degree, having higher chances of going extinct than poikilotherms. The reason why we think this reinforces our hypothesis is because endotherms and mesotherms, because they are homeothermic animals, they have higher metabolic demands than poikilotherms. And because of that, they need more food. And because of that, they need more area. Therefore, a loss of area is going to affect them more, as opposed to poikilotherms, for instance, they will be more affected by temperature per se. Now, in terms of the effects on functional diversity, we use an approach that's been used in ecology. We use the methods of Mouliot in 2013, in which you have a species pool. So we use a species pool we gather from the paleobiology database, and we assign these traits. So the categorical traits that we assigned were maximum body size, gill, that we call gill, or the type of diet, macropredator, herbivore, micropredator, and so on, habitat, zone, and thermoregulation. So we assigned these traits to each of these taxa, and we created a functional space based on a principal coordinate analysis. We also did some presences and absences, because in this approach, you create one single space, and then in that single space, you quantify, based on the presence and absences, the volume of two different communities, and you compare those volumes. In this context, a shift will be the lack of overlap between two functional spaces. We did that, and this is what we found. So this is the functional space for the Pliocene before the extinction and the Pleistocene after the extinction. Each dot here represents not a species or a genus, but a functional entity. A functional entity is a unique trait combination. For example, this is a functional entity that is right here, and it has a body size of more than 12 meters, is a macropredator, is in the pelagic zone, coastal habitat, and mesotherm. This particular functional entity is filled by one genera, and one species actually, which is Megalodon. We found that after this extinction event, we lost 7 functional entities, so 7 of these dots didn't make it to the Pleistocene, and also 4 evolved. Now, these plots here that don't take into consideration sample differences, but in fact, the sample size of the Pliocene and the Pleistocene were different. To cope with this, we did some bootstrapping, and we standardized the sample size of both, and we run a bunch of iterations. And we found that once you take that into account, there was a loss of 15% of functional richness or functional space, and also 20% of shift. So to summarize, we have that we lost 1/3 of of taxonomic diversity. We found that we lost 15% of functional richness and a 20% of shift. So these numbers may seem modest or not a big of a deal. However, when you compare this with other studies that have done similar approaches to assess ecological consequences of extinctions, we find that actually they're very high. For example, this paper by a colleague in the Naturkunde Museum found that after the mass extinction event of mollusks, you only lost 1 functional entity. So the reason why we think we have such a difference is because we're talking about marine megafauna. So these are big animals. And actually, because they're so big, they're not very redundant. So for each functional entity, we have, for the most part, 1 or 2 taxa filling that space or filling that particular group. So of course, when you lose one species, you lose functional entities. So we wonder if that's the case for mollusks. We couldn't do some sort of direct comparison with other studies because it depends on what traits do you use to do that, what resolution, etc. So in case of mollusks, this is the kind of the theory we're working on. You have a range of functions just with marine megafauna, but because they're smaller, they tend to be more redundant. So for example, if we take that these boxes here are functional entities, we'll see that for mollusks, we think, or the hypothesis, that we have a lot of species in each of these boxes. When you remove some species, you don't lose these functions because there's still at least 1 species or genera playing that function. But if you remove other 10 species in this community, then you do lose some functional entities. So we wanted to know if this was the case or this difference was evidence in this other extinction event. So this other extinction event I'm going to talk about is a well-known extinction event that occurred in the Caribbean. It happened around the Pliocene and Pleistocene as well. And it's been said that 70% of benthic benthic species went extinct, or genera, sorry. And why? Because when the Isthmus of Panama formed, the oceanographic conditions of the Caribbean changed, and this causes extinction. This is debatable now. So we gathered a huge data set of Caribbean mollusks, and we run the pyrite analysis. Indeed, this extinction took place in the Pliocene, Pleistocene with our methods, and we found that for mollusks, there's an extinction of 35%, only for mollusks. So it's very similar to what we found with the marine megafauna. Interestingly, or in agreement with our hypothesis, we found that changes in functional diversity were almost not seen. I mean, we were almost negligible. So when we see the changes in functional entities, it doesn't change much during the last 23 million years, and not during the extinction event. Same for redundancy, and same for functional richness, which is the volume of the space. So let's ignore these lines for now, and just this one is the volume. So as a home take message, what we found is that there was 2 extinction events. One was global, one was regional. In both, we lost a similar amount of genera. However, with the marine megafauna, we lost around 7 functional entities, and for mollusks, only 1 or 2. And we think that is because redundancy is a very important determinant. So for example, the redundancy of marine megafauna is 1 or 2 genera per functional entity, whereas for mollusks, we have at least 4. So we propose that functional redundancy is a key element in assessing the ecological impacts of extinctions. And with that, I would like to thank my collaborators, and actually for the mollusk part, I'm sharing first co-authorship with Christine Bacon, and thank you to my postdoc hosts. Thank you.
Any questions? Yes.
Hi. So you looked at extinction of mollusks, okay? Based on data in...
We collected the data. No, we collected this data.
Okay.
Yeah.
So did you look at abundance change? Because the primary shift that happened in that extinction in the Caribbean was not extinction of trophic groups. It's not really extinction necessarily of taxa, but it was a massive change in relative abundance of particular functional groups. So that's what happened, rather than it being a massive turnover in,
That happened, but we also lost species in genera during that extinction event.
Of course.
Yeah, we're not looking at abundance for now. We're just looking at presence and absence. But because we're using pyrite, this kind of takes into account that, because we calculate the time of origination of extinction of each taxa, and then we kind of fill up the gaps throughout time. So we're taking into account that, but not in the same sense of abundance. However, we do have the data set of previous studies that have been done then in Panama that those include abundance. So our next step is actually to compare that with the other data that was collected only in Panama and Costa Rica, which includes abundance, and see if we have a similar trend using our methods.
Any other questions?
Thank you.
It's always a tricky thing. All right.
Yeah, if you can click that anywhere, basically.
Sure.
More than it's actually, I guess, it's straight, so.
Okay.
Slides and points.
Is that the advance?
So next we have Lisa Park-Bush speaking on behalf of a number of co-authors on the estuary effect in the origin of lake form.
Okay, thank you, Mike, and thank you for coming. Today I'm going to talk to you about something that I've been interested in for a very long time, and that is the origin and maintenance of diversity within lacustrine faunas. And so my co-authors are Andy Bush and Michael Wren from University of Connecticut, and then 2 of my former students, Gary Motes and Timothy Astroff, and of course always acknowledge funding sources. So what I want to basically do is focus on a few questions here. First, a question that many people have asked is, how did non-marine, and in this case lacustrine faunas, originate? What clades actually invaded through time, and what effect did that invasion have on a clade or a phyla's diversity, and how often did these invasions occur? We also were looking at how clade invasion is related to ecosystems and ecosystem changes through time. And then related to that, how sea level change and plate movement influenced clade origination and diversification. So looking at this is a large macro sort of analysis using multiple data sources. And the genesis of this actually went back to when I was a very wee one as an undergraduate and graduate student. Jane Gray had published a large compendium in PaleoCubed on lacustrine faunas. It was highly influential at the time. And from that work, she published a sort of originations of lacustrine faunas chart, which you can see here. And so this is just first occurrence datum of major lake faunas through time. And so that kind of made the idea kind of possible. So when we looked at paleobiology database, fossil record 2, we also looked at Neotoma, and we started our own database of lacustrine faunas through time. These are lake basins through time, and we have about 3,500 references now, over close to 400 basins as reported today. So we have multiple data sources, and I also want to point your attention to this Hydrobiologia compendium in 2008, which was also really informative for learning about modern lake faunas. So if you look at the Metazoan tree of life, and this is Hellenic and Pasmanic, it's a little old, but for this purpose, it simply points out something very critical that is so compelling, at least to me, and that is what we did was we parsed out the differences between marine-only clades, or phyla, versus clades that they have a continental component. So we were calling them CAM for this talk, continental and marine, and MO, marine only. So the colors will be green for CAM and blue for MO throughout the talk. And so what is so compelling is this little fact here, or pointed data source. The number of species that are marine only is about 0.41% of modern standing diversity. And so the diversity, these are the modern standing diversity in parentheses. You might not be able to see it too well in the back. But so basically the take-home message is, for the most part, clades or phyla that can invade or have a component that goes into the continental realm, whether lacustrine or terrestrial, are far more diverse than things that can only stay in the marine realm. And so we think that that's fairly interesting, fairly significant, and it really does open some doors to some other interesting questions. And so one of the questions is, well, why? And we think it has a lot to do with how things osmoregulate. And so in 2007, we published a paper in which this figure appeared. And we think that for the most part, these marine-only clades, these things that can't or don't have a continental sort of subclades or anything that came into the continents, whether in aquatic realms or in the terrestrial, are more or less osmoconformers. Whereas osmoregulation becomes way more important when you're in all these different diverse conditions with respect to mainly salinity. So think about the density difference between a marine salinity versus freshwater or hypersaline, and you can kind of see why this then becomes sort of an important sort of, and the synapomorphies vary across the tree, right? So mainly it's the ability to regulate your oxygen content and flush out all your waste to allow them into different environments. And so the other thing we did was we looked across the tree of life. So this is phyla here, and this is the modern standing diversity that we showed you earlier. And we noticed something very peculiar, and that also supports this, and there is sort of this demarcation or salinity divide between things that are continental marine versus things that are marine only. And so there are exceptions, of course, the Chynodermata, Entoprocta, and the Onchophorans on the other side. But for the most part, clades, and in this case, phyla, that can go into a greater range of salinity habitats are far more diverse. And so we are calling this the salinity divide, and we think that this is a testable hypothesis for various groups. You could take basically any type of group and delve into this a little bit further. So we think there is this diversity disparity that exists between these two, the sort of the salinity divide between these two sort of CAM versus MO entities. I should mention here, very important, obviously arthropods are always an exception to everything, right? So if you take the arthropods out, this still remains true. So, so arthropods exclusive, this still remains to be the case. Oops. So, right. So we did take a particular group, the mollusk, and in this case just the bivalves and gastropods, and looked at this a little bit more in depth. And for the most part, the continental and marine mollusks, the CAM mollusks, are more speciose than the marine-only mollusk groups. This is at the superfamily level and also at the generic level. We also note that the clade origination, has been said previously, is predominantly in nearshore habitats. We've known that for a while. And also that taxonomic longevity is greater for the CAM or continental marine faunas versus the marine-only taxa. And the data that show that quite compellingly are these. This is genera. This is pulled from the paleobiology database and also some other sources from our own database. But this is so this is basically a survivorship curve. So this is percent surviving. This is 100%. That's 1%. So the longer the curve, the fewer survive. So in this case, the CAM bivalves have a higher survivorship than the MO bivalves. So in this case, we're positing that one of the reasons why you have greater diversity is also having to do with survivorship. Now, whether that survivorship has to do with being able to osmoregulate, we have not quite made that link. We also looked at the gastropods, and they seem to show the same thing. So again, here's total gastropods. This is survivorship. So percent surviving is 100% and 1% down here. This is our CAM gastropods. This is our MO gastropods. And here you can see higher survivorship for the CAM gastropods versus MO. Now, again, these are hypotheses that people can go out and delve deeper and look at in more detail and model things in different capacities. But we think that this is something that does explain some of our data that we're seeing in the larger study. So the other thing we wanted to look at is invasions. So we think that things are invading, and that this is one of the ways that things are establishing themselves in lacustrine environments. So what we're looking at here in this graph is just the phyla and the number of invasions, meaning that the number of things within, this is from our own data set now, the number of times we see something that is a first occurrence datum throughout the geologic record for an aquatic fauna that's in a lake. So this is the number of invasions, and this is the frequency of invasions. And the take-home message here is that invasions occurred throughout geologic time. So there wasn't just one invasion and then things radiated, but things were invading multiple times through geologic time. And oftentimes the things with the highest amount of diversity has the most invasions. We're not quite sure if that's artifactual, but we think that our data are holding on that. But this is something that we'll be looking at in the future. So for the most part, clays are coming in multiple times throughout geologic history. And once a species does come in, or once a base of a clay comes in, it stays in. So that's another kind of interesting thing that we've been able to see. So how are they doing this, and where are they doing this? So we think that for the most part, lacustrine faunas are originating through an estuarine gateway. So we know, obviously, that estuarine space has varied through time due to sea level fluctuations. And so the obvious thing that we're thinking of is that at one time, you had these large river valleys, and they're being flooded, and you're getting a large amount of invasion passively or actively. And then as sea level drops, you get things stranded in different varying salinities in these lakes. So sometimes it could be hypersaline or saline or fresh water. And so we know for one thing that within estuaries we have different adaptabilities. So we know that within an estuary we have sort of this salinity wedge, and that things can be actually adapted just into that highly variable space, ecospace within an estuary. We understand that. The linkage between that and actually clay diversification on continents is not quite known to us yet. But we know that estuaries have high influxes of nutrients. They tend to be less diverse than other aquatic environments. We know that we have specialized individuals. We know that there's high turbidity, which might impact their ability to invade. But we also know that multiple times throughout geologic history, we've had mass expanses of estuarine space. And so this is just a reconstruction of Ron Blakey here. This is just geologic time represented this way. And these are from our own lake database here, just showing you various places where our lake basins are associated through time. And the idea here is that estuarine space has increased and decreased based on sea level. So during times of maximum flooding, you have the greatest extent of estuarine space, and you have times when we've had ice-free worlds. We don't necessarily see a latitudinal gradient to diversification, so we're looking into that a little bit more, and that will be hopefully forthcoming. So finally, looking at sea level and origination, what we did here is we took the Hawk sea level curve, and instead of just plotting the curve, we basically just looked at the fluctuations around a mean, which is this big, this black line. So this is geologic time here. So you're either stable or you're fluctuating. So this is actually a plot of sort of greatest amount of fluctuation around the mean. And then this is our, it says originations, but really it's first occurrence datum for lacustrine faunas through time. And so this is, again, this is the mean. This is the standard deviation of the multi-point mean slope of the line of our originations. So here we're seeing that coincidentally originations were highest when you had highest amounts of fluctuation. So the highest amounts of sea level incursion, sea level fluctuations through time. So here in the Carboniferous, so down in Silurian or Ordovician, Carboniferous, it seems to not hold up in the Permo-Triassic, but it holds up in the Cenozoic and in various parts within the Mesozoic. So we think that there are times of maximum flooded continental areas that coincide with times of maximum originations, first occurrence datum. There does not seem to be a strict correlation between first occurrence datum and greenhouse conditions, which is also kind of interesting. So, to summarize, we think that continental faunas, and this we're mainly focusing on lacustrine faunas, established themselves through estuarine gateways, and they did so by multiple invasions and maximum flooding events when ecosystem space expanded on the shelf margin. We know that there's a diversity disparity between exclusively marine clades versus those that can adapt to continental settings, and that clades invading the continents did so by something we're coining as the estuary effect, and did so numerous times, and that global tectonic and geochemical cycling throughout the Phanerozoic influenced continental colonization, and hopefully diversification. We'll be able to see this in further studies through time. So thank you.
So sorry, no time for questions there. We'll see these later.
Just clip on somewhere sort of high up there. Do we need it? Really, it's
So
really
the
for
final
recording
talk this
actually.
morning
Oh,
is Chris
okay.
Spalding, who'll be talking about minimum population
Here?
extinction time driven by
Maybe actually.
stochastic environmental risks.
Sorry, technology. Right. Hi. So I'll try to keep this relatively short because I'm hungry. So by day, I am an astrophysicist, planetary astrophysicist, but I flew here from California, and because of the time difference, it's not daytime there. So I'm going to tell you about what I do at night, and that is population dynamics under fluctuating environments. And so we've already heard a lot about extinctions, a lot of very interesting stuff that I'm fascinated to hear more about. But we know that extinctions are happening all the time. They're critical for sculpting biodiversity. And just by way of illustration of that point, here's a plot from Bambach 2006, which essentially you can break up the Phanerozoic into lots of different horizons and ask how much extinction goes on in each of those horizons. So famously, there are the mass extinctions. For some reason, there are about 11 of them, 16 of them here or whatever. But the mass extinctions are just sort of a tail end of a distribution of extinction events. Every horizon has a certain number of extinctions, and just some are more than others. And so a question we can ask is it more important for sculpting biodiversity if you're a rare big event or if you're a really common but small event? And attempts have been made to do this using the fossil records, such as Raup in 1991, where on the x-axis, you have the kind of severity of the event, the fraction of things going extinct. And on the y-axis, we have the number of total extinctions over the Phanerozoic that those caused. And in general, it's the more frequent, lesser events that are causing most of the extinctions over time. And in some ways, you would think are more important. Now, of course, when you wipe out 70% of an ecology species or ecosystem species, that's going to have a different type of effect than if you wipe out 10%. But just in sheer number of extinctions, the common small events seem to win. And so this is merely an empirical curve, but we don't have a really full mechanistic understanding of how some of these patterns come about. And so that is my goal, to look at how the time scale, the rarity, the severity of extinctions will affect a population differently. And so in order to do this, I need to start modeling a population, seeing how long it takes to go extinct. And the easiest way to model a population is to take a deterministic approach. And this is where you define some carrying capacity and some growth rate, A, and the population will just kind of reach the carrying capacity and stay there forever. The problem with this model is that the population will never go extinct. It'll just stay at its equilibrium carrying capacity forever and not change. And so the models I'm going to be looking at are actually stochastic models, where instead of a birth rate, you prescribe a probability of a birth happening in a certain amount of time. And instead of a death rate, you prescribe a probability of a death happening in a certain amount of time. And where these two kind of average rates are equal, that's where you define your carrying capacity. But in reality, rather than the deterministic picture where you just kind of approach the carrying capacity and stay there, these models will go to the carrying capacity, here set at 120 arbitrarily, and these are just two different initial conditions, and they will kind of reach that average and then jitter around it for a while. But eventually, the key here is that you'll get a bad roll and you'll go extinct just by sheer chance. Like the pure dodo here. But, as we know, the dodo didn't go extinct because of pure chance. The dodo went extinct because of a very specific type of environmental perturbation, i.e. Homo sapiens, us. And so one thing that I'm going to add to the model is environmental variability. And so environmental variability has a lot of important effects on the time it takes a population to go extinct. And you can model it in a plethora of different ways, which I'm happy to discuss if you like discussing such things. But I picked a very specific type, or a very general, actually, type of stochastic forcing upon the population. And it goes as follows. I have a good state, a K plus, a high carrying capacity, and a bad state, a K minus, where the population kind of starts in the good state, stays there for an average time of 1 over alpha epsilon. So alpha is like a rate, and epsilon is just a number, which will hopefully become clearer later. But after that time, it flips into the bad state, which lasts 1 over alpha. So there's this number epsilon, which is kind of the ratio of the time you spend in the good state and the bad state. And I parameterize that bad state just by augmenting the death rate by a factor of A. And then sometime later you flip out, and the system keeps doing these jumps between good and bad state. And another pictorial representation of that is you have the flipping of the environment in the gray lines, and the population kind of tries to follow it, but it's still jittering around what the instantaneous average is. And so what you can do from these types of models is you can tune the alpha, the rate of the fluctuations, and then calculate the average time it takes this population to go extinct. And there are lots of different ways of calculating the average time this population takes to go extinct. There's a nice kind of semi-analytical mathematical technique that you can write down based on a technique used in the MacArthur and Wilson island biogeography books, if you know those. I don't have time to go into that, but the brute force way you can do it is to just code up a numerical trajectory like this in a computer and just run like 100 simulations and just note whenever each went extinct and average the number, and that gives you mean extinction time. And I like to be in sync with the animals that I'm studying, and so I use a computer program that's so out of date that even the dinosaurs would understand it, and that's Fortran. So that was not necessary for the talk at all, because I'm going to skip over all the methods and just go straight to the results. And so I'll walk you through this graph, because this is essentially the main point of the talk. So on the x-axis, I have the switching rate on a logarithmic axis. So that's the alpha that I showed you earlier. On the y-axis, I have the log of the mean time it takes the population to go extinct. And the crosses are the numerical solution, and the lines, the solid line is the solution that you can kind of write down by hand. And the fact they agree was nice. And so really the main thing to notice here is, well, first of all, I've picked alpha equals 1. So this is a model where you're spending equal amounts of time in the good state as the bad state. But as you increase the switching rate, your extinction time goes down. Things get worse and worse and worse and worse until at some key point, you go back up again. So how general is this pattern? I choose epsilon equal to 1, so equal times in good and bad. What if I make it more like what we'd call a catastrophe, where epsilon is less than 1? So if epsilon is less than 1, that means the bad events are rarer than the good events. And so as I make epsilon smaller and smaller, so 1/5 of the time, 1/100th of the time, this peak, this minimum, actually gets sharper. And I have a name for it. I call it the most catastrophic catastrophe, the alpha that minimizes your extinction time. So this is kind of a weird pattern. But the key is that because I prescribe my environmental forcing in a kind of intuitive sense, we have a feeling for good times and bad times, the talks and then the coffee break and then the talk. But we can actually understand it sort of heuristically. And so I'll just try to run you through this. So suppose we're on the small alpha end, so things aren't fluctuating much. The population will kind of just go extinct because of those initial fluctuations. So this takes a long time just for fluctuations within the population, so-called demographic stochasticity, to actually make it go extinct. But if your events are rare enough, that'll happen before anything changes. But then you get to a point where the bad event is happening sort of soon enough that it will make you go extinct. But because it's rare, well, I said it the wrong way. It'll hit you before you go extinct, but because it's so rare, it's really bad, and so it makes you go extinct almost every time it hits you. And so the extinction time goes down in proportion to the rate of those bad events. So if I hit you with an asteroid twice in 60 million years, you will die twice, rather than, rather than a smaller event that might not kill you every time it hits you. But then eventually you get to an event which is common enough and less severe enough that you typically survive them, and the extinction time goes back up again, until eventually you reach the really common but really just annoying things like rain and things like that British people love, where you just kind of see the average extinction rate, or the average death rate with time. So that's why the graph flattens off again. So I did this work during a summer school in Woods Hole, somewhere in Massachusetts. And I worked on it with a guy called Charlie Doring, and I believe he forgot that he actually discovered this in 1992. This is Charlie here. If I had a picture of me, I would have been a baby at this time. But there's something called resonant activation, and it's sort of simple. It's if you imagine a particle undergoing Brownian motion in a potential well, and that potential well is fluctuating up and down at a certain rate, and you change the rate of the fluctuation and look at the mean time it takes that particle to escape, then you find an exact same shape of curve that I found for the extinction. And this kind of isn't surprising, because an extinction is kind of like you're escaping the potential well of the living and going into the potential well of the dead, where you do not come back from, except Lazarus, tax, things like that. But anyway, so it's interesting that this was a previously discovered phenomenon. And essentially what's happening is that you're getting events, so the blue line are the bad events. You're getting bad events that are just enough for the population to make it through, but no more. There you've got the perfect balance of a severe event, but also one that's happening common enough for it to cause a lot of extinctions when it happens. That's the kind of qualitative depiction of it. So I'll skip that. So far, the augmentation in death rate has just been an arbitrary value A. I set it to something. So the next step is to look at sort of modeling real systems. We need to get a grip on what this number A is and the other parameters in the problem. And one example we've heard of is sea level change. There's sort of a species area curve that you can use to get numbers in this. But another one is something I've been working on regarding the energetics of whenever you undergo ocean acidification, how that may augment your death rate because of energetic constraints. And I have a poster on that tomorrow upstairs if you're interested. But I said the conclusions multiple times, but there's a most catastrophic catastrophe, which is the frequency that minimizes your extinction time. And I think this might have implications for evolutionary rate, which I didn't have time to talk about. But essentially, if you think of the population not as individuals, but as alleles, then you can minimize the time of an extinction of an allele, which is kind of like evolution. So I'd be happy to talk about that more, but thanks for listening and enjoy your lunch. That's fine, that's fine, that's fine.
So with that, thank you for your attention this morning. We'll have to hurtle downstairs and get there before the other people. P41 to the sandwiches.